The Cell, Chapter 4, Cell Structure. In this chapter, we have information about many of the common structures found in eukaryotic cells. We also have some examples perhaps of prokaryotic cell structure as well that are discussed in this chapter. But keep in mind these are common structures found in most types of cells, but does not represent the variations that can be found in different types of organisms that can be much more unique than what we'll see in this chapter. So these are the very common types of structures we'd expect to find in cells here on Earth. For the most part, for plants, animals, and fungi, the cell is the fundamental building block of an organism. We will find that there are entities called viruses that challenge this concept of what is an organism and does it require a cell structure. But for our purposes, when we're talking about multicellular organisms like plants, animals, and fungi, the cell is the fundamental building block. If we have a single-celled organism, that one cell is in fact the whole organism. In biology, we have a hierarchy to organize what makes an organism. So if we take a look at an organism, the very fundamental building blocks of an organism, again, we're thinking usually of a plant, animal, or fungi, something multicellular. The cell is the basic unit. When we assemble various types of cells that have different functions, then we might have tissues. Tissues are composed of multiple cells that have a particular function. And if you combine several different kinds of tissues, that each tissue has a slightly different function perhaps, then we form an organ. An organ can be composed of multiple bits and pieces of different kinds of tissues. Those cells are working toward a common function, and those organs can be collectively organized or assembled into what we call an organ system, like the respiratory system, or the lymphatic system, the central nervous system. Multiple systems that function together form an entire plant or animal, or what we call an organism. Now, will we look at fungi and find those same kinds of hierarchies? No. If we take a look at fungi, when we get to looking at cell structure and tissues and organs, we really don't see some of the unique organ structures in fungi that we would expect to see in a plant or animal. And so that's what's going to distinguish something that we're going to define as a fungi from a plant or an animal. Fungi do not have unique organs that are composed of different kinds of tissues. So when we take a look at this hierarchy of plants and animal body organization, it really is just for those two major categories of organisms. Cell sizes vary, and most cells are too small to see with a naked eye. There are exceptions. There are some cells that you can easily see without any additional magnification. But for the most part, cells are too small for us to see with our unaided eye. There are two general categories of microscopes, well actually three now, that can be used to view these tiny little cells. When we're looking at cells through a microscope, there are two characteristics that are very important for us to have the best image or view possible of the cells that we're looking at. And the two parameters that are most important in microscopy, which is the use of a microscope to view material or items, we have to have magnification, which means enlarging the image, and good resolving power, or what we call resolution, which means a nice, sharp, crisp, clear image that defines or distinguishes tiny, unique pieces and parts of whatever we're looking at through the microscope. So the image on this screen gives us a good idea of magnification because these are examples of what we call diatoms. You really can't see these diatoms in a drop of pond water with the unaided eye. And of course this photo, we see some nice clear images of what each individual diatom appears to be. That's good resolving power. And we need both of those characteristics in a microscope to give us our best image of whatever we want to look at. So here we have an example of magnification. Notice that when we're magnifying something, we're increasing essentially the image and getting closer and closer. Every time we zero in and enlarge an image, the resolving power is going to be critically important. So it's not enough that we just enlarge the image. We also have to make sure the resolving power is matching or keeping up with that magnifying power. So the resolving power is the ability to see two adjacent structures as separate. And the higher the magnification and the better the resolution, the more clear the image appears to be. We see on the left what we call a light microscope image of an organism called a radiolarian, and on the right an image using a different type of microscope that we call an electron microscope, also giving us a nice sharp clear image. They're both at the same level of magnification, but the two different microscopes with the same magnifying power with slightly different resolving powers give us each information about the structure of this radiolarian. So we would combine these bits of information so we could understand what a radiolarian really looks like. One general category of microscope that we commonly use, and especially in the teaching laboratories, are the light microscopes that use visible white light to view an object. There are magnifying lenses, usually made of glass, in these microscopes, and for the most part there's two or more magnifying lenses in the microscopes that provide magnification. These lenses are also aligned to a particular arrangement, so we have good resolving power as well. Now when we take a sample of cells from most organisms, like plants and animals, they tend to be transparent. They don't have any color. If we were to look at them through the microscope, especially a white, or what we call bright field microscope, the bright field means we're using white light or visible light to view that item. And if we look at something that's not stained or has any kind of contrast, it's very difficult to see these materials through the microscope. So as the image on the lower left shows you, oftentimes we're going to use some kind of stain or dye to highlight the features of whatever we're viewing through our bright field microscope so that we can distinguish all the different parts. We can see them with the stain that provides the contrast. There are other types of microscopes that use visible white light as a source of illumination, but they have slightly different configurations that alter the pathway of the light so we can still get some contrast to our image, but it does not require a stain. And that example is on the right hand side in what we call a phase contrast microscope. With the phase contrast microscope, we can take a sample of cells, which is illustrated in the photo on the right. We don't have to stain them, but because the light has been altered in its pathway around the specimen, we now can see more detailed structure. There are various different types of microscopes that use visible light in different configurations, and they all have their advantages depending on what we're trying to see or distinguish through our microscope. A second category of microscopes much more sophisticated and expensive, are the electron microscopes. Electron microscopes come in two types. With an electron microscope, we're using electrons instead of visible white light to view something. And there are one category of microscope that can use an electron. It's called the transmission electron microscope. Transmission means just that. Transmission means passing through. So we're transmitting electrons through the specimen, which is illustrated on the lower right. The lower right is a cross-section of a capillary. That's the white area that you're seeing. And the little black thing that looks sort of like a funny-shaped C on its side, that's a cross-section or cutting through a type of cell called a red blood cell or an erythrocyte. Capillaries are the tiniest blood vessels in animals that simply allow one single red blood cell passing through at a time. And so transmission electron microscope means we're passing electrons through the specimen to reveal some kind of structure of that specimen. A second type of electron microscope is the scanning electron microscope, which is illustrated in the bottom left photo. The scanning electron microscope takes a facsimile of the specimen, so there's a little bit of processing needed for the specimen for both types of electron microscopes. And with a scanning electron microscope, we're essentially revealing the structure by passing electrons over the surface. We're scanning the surface of the specimen. Students often prefer to view scanning electron microscope images because they have a more three-dimensional appearance. So when we take a look at the photo of the red blood cells, also called erythrocytes, in the photo we can see sort of the structure, the overall appearance of the erythrocyte. Now, scientists will combine the information from both types of electron microscopes. We get an idea of what the internal structure would be from the transmission electron microscope, often abbreviated TEM, and we'll combine it with the information discovered using a scanning electron microscope, abbreviated SEM. So we can get a true idea of what the cell really looks like and perhaps even its function. A third category of microscopes that we often forget to mention to students are the scanning tunneling and atomic force microscopes. With the atomic force microscopes, we're not using visible white light and we're not using electrons. Instead, we're using a tiny little, we call probe, that glides over the surface of a specimen, and then we're going to be using laser beams and photodetectors and computers to interpret that flexing of the tip of the probe, which is illustrated on the right in green, over the surface of a specimen, so that we can reveal the actual arrangement of atoms in a specimen. So atomic force microscopes allow us to view at some of the tiniest scales ever imagined of the structure of particular specimens. And yes, AFM, or atomic force microscopes, have now been used to view biological materials as well. So it's been very interesting to see and reveal the differences in structure and function of different pieces and parts of cells using these three different categories of microscopes: the light microscopes, electron microscopes, and the microscopes that can view right down to the level of atoms.